Welcome, everybody. Thank you for joining our sixth annual virtual event of the Therapy Acceleration Program. My name is Blaine Robinson. I'm the Vice President overseeing our Therapy Acceleration Program here at Blood Cancer United. Today's focus is on TAP's role in supporting biotech companies developing much-needed innovative therapies for blood cancer patients. Over the course of the next hour, we will hear from three of our TAP partner companies. But first, I wanted to give you a brief overview of TAP before passing on to my colleagues to lead the discussions with our panelists. At any point during the webinar, please feel free to submit questions to our panelists via the Q&A feature, and we'll attempt to answer them at the end of the webinar. I also wanted to let everyone know that we're recording this event, and we'll be making the event recording available later next week. So TAP, our venture philanthropy initiative here at Blood Cancer United, was established in 2007. It was really one of the first venture philanthropy programs in the biomedical space. Our primary goal is to support the mission of Blood Cancer United to find cures for patients. Unmet medical need is what drives us to invest and make a difference, and no indication is too small. The program has invested over $115 million to date, and there are over 30 active clinical studies ongoing. We're actively investing as we speak, and we're very proud to say that our funding has contributed to 6 approved therapies. Among them is Vyxeos, a novel formulation of cytotoxic therapy for AML patients with a very particularly poor prognosis. It is also now available to pediatric patients. Yescarta was one of the first approved CAR T-cell therapies and the first one approved for large B-cell lymphoma. Elzonris is a really good example for one of the approvals of a very rare indication of blood cancer patients, blastic plasmacytotic dendritic cell neoplasms. And TAP has been instrumental in helping to expand indications for agents that are already approved, and a good example of this is Copiktra, a dual PI3 kinase inhibitor, which has already been approved for several blood cancers. However, our work contributed to a clinical trial that supported the inclusion of this agent in the NCCN guidelines for peripheral T-cell lymphoma, and more recently was given approval for cutaneous T-cell lymphomas. TAP conducts equity investments as part of a syndicate in both private and public companies, focusing on companies that are developing truly novel, potentially groundbreaking therapies. These mission-driven investments are not driven by financial returns like traditional venture capital. However, we've had success and have received over $110 million to date that's helped fuel additional activities to support our mission and more recently to help grow TAP. Several of our partners have had significant strategic transactions. Just recently, last month, one of our partners, Imcheck Therapeutics, was acquired by Ipsen for their promising therapy being developed for AML. Importantly, as part of our investment, there's a collaboration aspect with our partners that helps us work with the company on making sure that the development of these agents can provide maximum benefit to patients. Shown on the right side, here we give you a snapshot of companies who have partnered with our TAP program. And I just wanted to highlight we're pretty comprehensive, including agents at all stages of development, from preclinical on the outside ring to phase 1 to all the way to registration-directed trials, focusing in all blood cancers, including, of course, leukemias, lymphomas, and myelomas, as well as myelodysplastic syndrome and myeloproliferative neoplasms. Boxed in blue indicate some of our recent partnerships. Now, I want to speak for a few minutes about TAP's role in our latest FDA-approved therapy, Komzifti, otherwise known as ziftomenib, a menin inhibitor developed by Cure Oncology. So Blood Cancer United has supported research in the development of menin inhibitors for nearly 20 years. We funded key academic research starting in 2005 that helped discover the role menin plays in the context of leukemia development, initially in MLR-rearranged leukemias, an aggressive form that strikes infants, children, and adults, resulting in poor outcomes, and more recently in leukemias driven by NPN1 mutations. Both types account for nearly 40% of patients with AML. The story of the first menin inhibitor began at the University of Michigan, where Dr. Grambrecka received several Blood Cancer United grants that began and sustained this exploratory work while also supporting her as a young investigator when pharmaceutical interest was minimal. During this time, TAP entered the menin story with one of our longtime colleagues, Jim Casper, leading the project for TAP. He currently runs our translational research program, but he was really instrumental working side by side with Michigan investigators, consultants, and other colleagues. From 2010 to 2014, critical and significant funding from TAP went to the University of Michigan to design and develop potent new compounds that could safely and accurately target the menin protein. The Michigan group was the first to publish about menin inhibitors, creating excitement, and biotech companies began to start taking notice. What happened next was our former chief scientific officer, Lee Greenberger, he called this Cura oncology CEO in 2014 and spoke about our pioneering work that we funded at Michigan. So once biotech was involved, this all set off a race to develop better molecules that ultimately could be tested in humans, leading to ziftomenib, which was the first menin inhibitor to begin clinical trials in 2019. Other companies quickly followed, and recent late-stage clinical trials have reported encouraging complete response rates in patients with relapse disease, and this has led to several FDA approvals in this space. Ziftomenib received FDA approval this past November, making it the 6th TAP-supported therapy to receive regulatory approval. Our commitment didn't stop when companies were testing their compounds in trials. We continued to accelerate the translation of menin inhibitors into viable treatments by working with biotech companies through our master clinical trials, BEAT-AML and PETAL. The BEAT-AML was the first comprehensive precision medicine trial for AML, where patient samples are profiled at diagnosis and patients are rapidly matched to targeted therapies. Several years ago, BEAT-AML partnered with Syndax, a competitor to Cura, to conduct a clinical trial to explore their menin inhibitor called revumenib in combination with standard of care agents in newly diagnosed AML patients. The BEAT-AML trial provided critical data needed to support a registration-directed trial that Syndax performed to expand revumenib for use in newly diagnosed AML, and this trial is now ongoing. Through PETAL, we've extended our precision medicine strategy into childhood leukemia, creating an international network designed to give children access to novel targeted agents. Menin inhibitors are a very high priority, ensuring that when we have promising adult trial results, these can be rapidly translated into pediatric settings. So ziftomenib is now included as a treatment option in the PETAL initiative, making it the first ever global pediatric trial for menin inhibitors. So what does TAB look for when we make investment decisions? We really focus on companies and programs that can make a dent in the treatment landscape. We're not just looking for what's out there today, but we're also looking for the unmet needs that are emerging. Even with new approvals, sadly, we still see relapses, and there remains a big need for novel agents. So for our process, we carefully look at companies developing true innovation first that have the potential to address gaps in the current treatment landscape. Our due diligence process is stage-gated, outlined here, and we take advantage of our ample connections in both the academic, scientific, and clinical world, as well as in the business world, to help us support our comprehensive diligence process. We're not just looking at the science and the clinical development plan, but also at the seasoning of the company, the management team, intellectual property, and their finances. Now today, specifically, it's a real pleasure that we'll be speaking with some of our partners who are in late-stage clinical development and are developing emerging new therapies for patients. We have a worldwide reach, and 2 of the partners today are even based in Europe. Before I bring out the first panelist, I would like to share our latest TAB video, developed in collaboration with Cabin 3 Media and generously supported by Kiki and Tui Connell and the Butler Family Foundation. So let's roll the video.
I was diagnosed with acute myeloid leukemia in 2021. It certainly was scary as I had little hope and I just didn't know what to expect. My parents and I were given the opportunity to be part of a clinical trial, which was Vyxeos, which is funded by TAP.
Our doctor was very pleased to find out that she actually was randomized to the trial side, and the very next day she started her treatment with Vyxeos.
Vyxeos is, I would say, my proudest moment, where we really were able to use nanoscale liposomes to deliver a drug combination for the treatment of acute myelogenous leukemia, or AML. And this drug significantly improved survival of AML patients compared to the current standard of care.
She actually had a very fantastic first round of treatment. She had very minimal side effects and was actually in remission. However, as a part of the clinical trial, we found out that she had a genetic marker that was almost a 100% indicator of relapse within a year.
I had to switch to the standard care of chemotherapy because I had to have a bone marrow transplant.
Everything was much more challenging. Just having to watch your child suffer and knowing that you were doing everything that you could to make her better while watching her get really sick.
I was 13 at the time, but children who are younger should never have to endure pain under that circumstance, even when they're fighting cancer. So I think that it's very important that Vyxeos had a lower, less extreme side effects than other chemotherapies.
What's most exciting about Vyxeos, after 5 years, the survival rate had improved for Vyxeos more than 200% compared to the conventional treatment.
We have had the best possible outcome. As of now, she is 4 years in remission.
I am now a senior in high school and I'm back doing everything I love. I would say that I'm cured. I'm cured of being scared. I'm cured of not having any hope. I'm cured in the aspect where I'm healthy, where I wasn't 4 years ago.
We feel like Lila's story is truly a story of hope because of TAP and all of the work that they do and all of the funding that they provide for all of these clinical trials. They are providing so much hope for families in positions just like ours.
Without the partnership support of TAP, I often question whether Vyxeos would have ever gotten approval. And because of that partnership, lives are currently being saved every day.
Wow, it's an awesome video. So I really hope you enjoyed the amazing video that was put together about Lila's journey with Vyxeos and the importance of clinical trials. So without further ado, let's hear how TAP today is working with biotech companies to tackle blood cancers. As a reminder, please submit any questions you have to our panelists via the Q&A feature, and we'll attempt to answer them at the end. So first, let me welcome our Chief Scientific Officer, Laura Grunbaum, to join, and she will kick off the first discussion.
Thank you, Blaine, and thank you to all of you online for joining us today. So as CSO, I have the great privilege to oversee our full portfolio of biomedical research activities. Many of you probably know that for many, many decades, we have supported blood cancer research and development through academic grant programs. And Blaine just mentioned the Beat AML master trial as well. And it's really one of my goals to strengthen and accelerate innovation in the blood cancer space by making connections between all the different components of our research portfolio and our many additional activities, our many mission activities. And an additional important piece that we're highlighting today is, of course, that we want to actively work with all the stakeholders in the drug development ecosystem. We think that is essential for us to really move things forward as quickly as we can. And without further ado, I'd now like to invite Jan Fagerberg, Chief Medical Officer of Enterome, to join me. Jan, great to see you. Thank you for joining us. So you bring literally decades of experience in immuno-oncology to the table. I think you've spent time in the academic environment, among others at Karolinska Hospital, before moving into the pharmaceutical industry. I think you were at Roche for a time, which I was as well in the past, and you had several CMO roles before joining Enterome. So we'd love to hear a little bit more about your interest in what Enterome is doing and what the company is up to.
Yeah, thank you first for having me. This is, of course, for our company, a great opportunity to work with you and to be in this webinar as well. And you have already told about my background, and it's I have worked in the pharma industry now since 1999, so it starts to become some time. And I had almost stopped when I found Enterome. And this was back in 2019. Enterome was founded already 2012 with the idea to utilize knowledge about the gut microbiome for different purposes. But what I learned back in 2019 is about a concept called the oncomimics, which is a very novel and unique concept which actually has as the goal to expand as fast as possible T cells that can kill tumor cells. And this is a general concept applicable both to solid tumors and hematology. So this was what attracted me to Enterome, and I think in the end that's why BCU are interested as well because of this concept.
So you mentioned the oncomimics, and this is a very unique concept. I think many people in our audience are familiar, of course, with cellular immunotherapy and other mechanisms, but there's something very different about the oncomimics. Can you explain that in a bit more detail?
Yes, so the oncomimics, this is something that is completely different from whatever you have heard about different T cell approaches, CAR T cells, TCR, transplanted things. But the oncomimics, in principle, are short peptide sequences normally produced by the gut bacteria. They are highly antigenic, and the key is that the ones that we select, they closely resembles targets on tumor cells. And these oncomimics, the one other key is that they can expand and activate tumor killing T cells, which we already have in our bodies. So it's an in vivo approach to actually get huge numbers of specific T cells. This is not we have it, you have it, I have it, these T cells in our bodies, because they are there to recognize bacteria if the bacteria we have in our gut would invade us. So in the gut, the bacteria help us with a lot of things. They are essential. But if they would be bad and invade us, then we have a T cell system to protect us. And these T cells are very efficient killers, some of them. And what we do with the oncomimics is that we expand these already existing T cells. And if we have been smart when we select these peptide sequences, we have a composition of peptides which can expand T cells that specifically can kill B cells. And that is our goal when it comes to lymphoma. So that we have created a compound that for lymphoma to specifically recognize B cells, and we call this EU2463. So we don't have a name yet, but this is what I will refer to. And it's a mix of 4 synthetically produced peptides. So it's very simple production. But the sequences come from the gut bacteria proteins. And these peptides can expand killer T cells targeting 4 B cell markers. So CD20, it's the same target as the well-known rituximab. It's CD22, CD37, and BAFF receptor. And we have created this with 4 targets to actually have a bigger chance to hit the tumor cells and also to counteract if the tumor is smart and try to escape, that we have multiple targets. And this drug, EU2463, then it's a very simple process. We inject this subcutaneously and together with an adjuvant to get a little depot effect, that it stays in the skin a bit longer. And the standard schedule is that we give 4 injections with 2 weeks apart, and then that's the key part. And then we have booster doses every month. But this all is a very simple and off-the-shelf thing. So it's nothing that is specific for an individual patient. Now, these peptides are specific for transplantation antigens. So we are now targeting patients who have the so-called HLA-A2 allotype. That means that it's 50% of us. We can, in principle, create oncomimics for any haplotype, but we, of course, with development, we started with the group that is the largest, and then we can expand on this when we have proven effect. So this is, in short, in a simplified way, how the oncomimics work. And the key, again, is that we expand T cells that are already present in the body, and help them to be activated, become many, and to target tumor cells.
This is a fascinating mechanism. I mean, it sounds to me like you're pretty much repurposing T cells that really had a different purpose in the context of the gut, and now you're repurposing them because there are so many different, I imagine, bacterial peptides that you can always find some of these memory cells that will also cross-react with the peptides on the tumor, right? So
Yes.
this is a fascinating mechanism. And I know that you are actively developing your lead candidate, in particular for follicular lymphoma. So can you tell us a little bit more why follicular lymphoma? What's the unmet medical need for patients, and why do you think this candidate is particularly well positioned to benefit these patients?
Yeah, so when we developed this compound, EU2463, targeting these four B cell markers, of course, we discussed with a lot of experts, both in the US and Europe, on what are the unmet needs, where can we place this? And the study we have been running, and which is the key, because for the development of this compound, is that we actually selected to test the compound in multiple indications in the same study. So, and that we would start with indolent non-Hodgkin lymphomas with the focus on follicular lymphoma, of course. So we have one group that is for patients with relapsed disease, and then we didn't dare to use EU-2463 monotherapy, so we do it in combination with lenalidomide and rituximab in relapsed refractory disease. Then we have one group of patients with newly diagnosed disease, but with low tumor burden and in need of treatment. So there we test a combination of EU-2463 and rituximab. But the real key, and this is what we have heard from everyone from the beginning, that is a compound like this that it was from the beginning assumed to be very non-toxic and have activity, would fit very well in what is called currently the watch and wait setting, where there is no established treatment. So this is the third population where we are testing the compound. And then it's, of course, monotherapy, no other drug added to it. And this is where we think that the unmet need is as highest. We think that it has a place also in the other settings I talked about, but that this watch and wait setting, both for patients and physicians, is a rather disturbing situation, because you have disease, and even if it might take a longer time before the need of more toxic therapy, it will in most patients come. And that's where we think that this EU-2463 monotherapy has the best fit, and because of the low toxicity from the beginning, it was a hope. Now we have shown that the safety is really good. And the hope was also for efficacy from the beginning of monotherapy. We have shown that there is efficacy now. So what we target as the main group is actually the watch and wait setting monotherapy, to make an impact for patients, that we can prolong the time until more toxic therapy, or in the best case, avoid it at all. So this is the group we really think would benefit mostly to start with. We think the other groups are important as well, but that's where we start for the next generation of studies.
And you mentioned it a little bit, but you've reported some really promising data over the last year at conferences, most recently at ASH. So can you maybe just very briefly tell us where you are and also what you're planning in the next step of clinical development?
Yeah. So during the last year, actually in December and at the Lugano meeting mid last year, we have reported data from all these populations I mentioned. And if one take the conglomerate of the data and talk about the general conclusions of this compound, that is, first, it is really, as I have mentioned already, really well tolerated. In principle, the only toxicity we see, that is local skin reactions, erythema and indurations. All other safety things we see, that has to do with the combination partner. So when we give monotherapy, it's really an easy treatment. So that's number one, the well tolerated. The second is that our intention is to expand fast and to huge numbers specific killer T cells that recognize these B cell markers. And this we have shown in all settings now that we really do this. The number of T cells that we expand, that we can measure in peripheral blood, it's many percent of all T cells. This is much more than you see with when you have an infection, influenza, for instance. So it's really good numbers. And now we have learned that they stay also in the body at high numbers for a long time. Our longest followed patient is now out 3 years after the last dose of EU-2463, and she has still a high level of specific T cells protecting her then hopefully from a relapse. She had relapsed refractory disease that went into complete remission on the combination therapy. So, and then we have learned that these T cells that is created and expanded, they correlate with efficacy outcome, especially if it's a fast expansion of these T cells. We know that then it's correlated with complete remissions of the tumor. So these are 3 things for all of the settings. Then in relapsed refractory disease, together with lenalidomide rituximab, we have a complete remission rate of 60%, also in high tumor burden patients with POD24. So this is the cohort we are still running in this initial study, and we are now optimizing the schedule a bit. So we hope to actually increase the CR rate. We will see. Already 60% we think is a pretty good for an add-on therapy where we don't make it more toxic of the R square. Then in low tumor burden patient with rituximab, it's a small feasibility cohort where actually 6 of 6 patients responded with 5 complete remissions. But that's a small number. The most important, again, is what we see in the watch and wait setting monotherapy. And there we have approximately a 50% objective response rate, but where also patients on stable disease are important, because we know that in more than 80% of the patients, we actually induce these specific killer T cells. So even if the tumor doesn't disappear completely, we think that this stabilization of disease can be very prolonged as well. So this is
that's really promising, and I know we would love to discuss it more, but I want to make sure that we give everyone an opportunity. And so I'll thank you here. I'm sure there are going to be more questions coming later from the audience. Thank you very much, Jan.
Thank you.
And now I'm going to ask my colleague Jun Shu to please turn on her camera. And
thank you, Laura. My name is Jun Shu. I'm the VP of Business Development and Scientific Strategy for Research. I am a 14-year veteran of Blood Cancer United. Today, I'm very privileged to invite the Chief Medical Officer from Faron Pharmaceutical in Finland. And hi, Petri. Thank you for joining us today. Really appreciate that you're taking the time, Friday evening, local time, to join us. So the TAP team knew Faron since the JP Morgan Week in 2021. So that's more than 5 years ago. So in June of 2022, TAP made the first investment in Faron. Then, which was quickly followed by a bigger second investment in January of 2023. So relatively speaking, we the TAP team knew the Faron, team a little bit before you joined the team. So you joined the team, the management team, relatively recently. So what prompted you to make the decision to join the Faron team as a Chief Medical Officer?
Thank you, June, for the question, and actually also for the kind invitation to this panel. My name is Petri Bono. I'm Chief Medical Officer at Faron Pharmaceutical. And by background, I'm an MD PhD associate professor at Helsinki University and also an eager clinical and translational researcher. Before joining Faron, I've been in various leadership positions in academia and industry, including as director of the Comprehensive Cancer Center at the Helsinki University Hospital for 7 years. And before joining the company, I was also the global PI in the first-in-man bexmarilimab trial in solid tumors. So I was familiar with the company before joining also. But really what drew me then to jump over to Faron was actually the science. And Faron is a clinical stage publicly listed pharmaceutical company that is focusing on tackling difficult to treat cancers through precision immune therapy. It's been founded actually already in 2006 as a spin-out from the University of Turku, where actually still the headquarters is located here in Finland. And we are listed in AIM London since 2015 and on NASDAQ First North Helsinki since 2019. At Faron, today, our clinical development efforts, they are centered around bexmarilimab, the first-in-class fully humanized IgG4 antibody targeting Clever-1 receptor that is a novel macrophage checkpoint. And by inhibiting Clever-1 by our monoclonal antibody, we are able, with bexmarilimab, reprogram these immunosuppressive M2 macrophages into more pro-inflammatory immune activating M1 type macrophages, thereby promoting anti-tumor immune responses. And although our initial clinical development was focusing on solid tumors, where we have generated data from more than 200 patients, demonstrate that single-agent Bex is well tolerated and shows promising activity. So we've been, since 2022 expanding our focus to hematological malignancies, where actually the biological rationale is particularly compelling. In solid tumors, with BEX, we are targeting macrophages within the tumor microenvironment indirectly to affect their tumor growth, and already there shown clinical activity. But in contrast, in myeloid malignancies such as AML and myelodysplastic syndrome, MDS, we have dual mode of action, since Clever-1 is expressed not just in the tumor microenvironment, but also directly on the myeloid cells, the blast cells. And this really provides a very direct therapeutic approach. So ultimately, I joined probably Faron for the same reason why TAP has invested on Faron, because BEX has truly huge potential for fundamentally to change the outcome of patients having higher risk MDS.
Thank you. We're so glad you joined the team. And then also personally, I'm very happy that we were able to meet face-to-face at ASH in Orlando in December. So to really help our audience, so I'd like to really dive a little bit more into the detail of the data and the clinical finding of Bexmarilimab in our HEMONC indications.
Thank you, June. Very happy to expand on this. So without BEXMAB that is targeting this Clever-1 receptor, that is a scavenger receptor highly expressed on these immune suppressive macrophages and malignant myeloid cells. So we've been able also to show that BEX reduces antigen presentation, impairs T-cell activation, and causes immune suppression. And really, we have been able to show in our patients in the BEXMAB trial that we can reprogram these macrophages from immunosuppressive to immune stimulatory, and really restoring the immune system's ability to recognize and clear malignant clones. So we've been able to show that we have a fundamentally different and novel way of reshaping tumor microenvironment. And then to the ASH findings, we've been presenting data at ASCO meeting, at ESMO, about more translational results, and then the newest update on the efficacy at ASH in December. And we presented their data from 55 MDS patients that were treated in our BEXMAB phase 1/2 trial, and the results were clinically very excellent when you think about these hard-to-treat, higher-risk MDS patient populations. We had in our patient material, very typical in clinical trials, highly enriched with very aggressive patient demographics, like 40% TP53 mutated patients, 47% were very high-risk category patients, but still we saw very encouraging efficacy rates. If we start with the complete remissions in the frontline patient population, 45% of the patients received a CR response. This is more than double what you would expect according to meta-analysis. With single agent AZA, you get 16 to 17%. With AZA plus BEX, we got 45%. This is one of the highest reported in any clinical trials in these frontline patient populations. In addition to the high CR rate, 85% overall response rate, and on top of that, within the CR patients, we had 67% of them also became MRD negative, meaning that no residual disease could be even with very sensitive measures detected from the patients. Very important for the patients, 57% of the patients who were transfusion dependent while entering the trial became transfusion independent. These were frontline results. In the relapse or refractory setting, we saw very long overall survival, 14.5 months, and it's 5.6 months according to meta-analysis that one would expect these patients, unfortunately, only to live, so more than doubling that overall survival. We also saw that clear immunological activation based on translational studies, and we were really able to show that bexmarilimab together with azacitidine works very well both in the frontline setting as well as in the relapse setting. It was well tolerated, didn't increase the adverse events compared to single agent AZA, and we were able to validate the very promising earlier preclinical findings that with BEX we can sensitize cells that are resistant to azacitidine, we can sensitize them back. So now we see very nice efficacy in patients. And really now we are at the point now that this efficacy, so it strongly supports moving forward to late stage registrational development with our bexmarilimab. And this is well also aligned with FDA. We had an end of phase 2 meeting last August with them, and we were well aligned with the development path forward with the FDA.
Okay, thank you. So okay, so really, we joined the, like we collaborate with Farron because really unique the mechanism of action, and also we're very excited about really the efficacy and also the well tolerability. So MDS patients, especially the high risk patients, they really need better, more efficacious, well tolerated therapies. So we really look forward to hear more progress, because you already talked about you are moving forward to next phase, which is great. So we look forward to hearing more great news and great progress from the company. But before I let you go for this discussion, I'd like to ask, you can whether Bex can benefit other patients beyond MDS, other blood cancer indications, or even beyond blood cancer, like solid tumor, and maybe with a very different approach. Because we understood you did solid tumor before, but now maybe you have a new, more specific, defined approach in solid tumor.
Yeah. Yes, there's certainly a lot of potential with BEX. In the phase 1 trial, we had also AML patients, and there are also activities that we are planning within AML. Due to the lack of time, won't go into details, but I want to touch a little bit the solid tumor field, because we're going to use BEX in combination with chemo and with anti-PD-1 antibodies in solid tumors. We have actually released that the first solid tumor combination trial, so it will be done at the Royal Marsden Hospital in London, where PD-1 refractory metastatic melanoma and metastatic lung cancer patients will be treated together with BEX. So BEX plus anti-PD-1, and we hope to show that we can overcome the PD-1 resistance by adding BEX to the treatment. And there's a good preclinical rationale for that. And this now is something that is about to be launched there. We also actually last week published that we're going to start another solid tumor combination trial, which is together with chemotherapy, because there's also nice data published 1 year ago or so at Nature Communications about synergistic effect of BEX plus chemotherapy. So we're going to do frontline metastatic soft tissue sarcoma trial at Vall-d'Hebron Hospital in Barcelona, one of the leading European sarcoma sites. So very exciting times also in the solid tumor field, but of course, priority number 1 to push forward the development of bexmarilimab in the treatment of higher risk MDS patients.
That's great. Unfortunately, we reached the end of our allocation for this time. So I will ask you to sign off for now, but don't go away. Blaine will ask you to come back. And so now for the next, the final panel for this event, instead of a TAP staff, and then we have a special guest, Carol, and she's our national board member. She's also a member of a TAP committee. And so today we have really a special treat. So Carol, so please just say a few words about yourself for our audience.
Thank you, June. I'm so pleased to be here. I am Carol Gallagher. I spent over 35 years in the biopharma industry as a operator and drug developer, and then for 10 years as a investor for a venture capital firm called New Enterprise Associates that invests in biopharma. And so now I've turned my attention to more philanthropic efforts. And I'm very pleased to have joined the board of Blood Cancer United, but also the TAP committee that oversees the TAP investment groups. And venture philanthropy, I've come to understand, is really a quite unique opportunity for your philanthropic dollars because TAP has had now a great history and shown that their investments have a return. And so if you think about it, your philanthropic dollar then can become many, many years ahead an opportunity to continue to be recycled into future investments. And of course, today we're very pleased to highlight a few of these TAP investments, and you can see how it is supporting our mission to bring innovative therapies to patients, but also that these philanthropic dollars will create a return on those investments, and then those dollars can be invested again in future innovation. So very exciting to be part of this.
Okay, thank you, Carol. Before I go, so I just want to remind the audience, please use the Q&A feature. We already have several questions already answered. So now I leave it to you. So see you. Thank you.
Thank you, June. So now I'm pleased to introduce Dr. Tina Albertson, Chief Medical Officer at Caribou. Welcome, Tina.
Thank you,
Tina.
Oh.
Yes, great. Thank you. Tina is a pediatric hem-onc in her training, and interesting relative to today's discussion, she was involved in the very early days of cell therapies for patients. She led the clinical trials at Juno for the approved treatment today, the CAR T treatment now known as Breyanzi. At Caribou, she's developing that next generation of cell therapy innovation. And I think you'll be excited to hear today from her about this technology and also the data thus far. Could you tell us more, Tina?
Yeah, thank you, Carol. Thank you for letting me talk about our work that we've been able to do in part because of the TAP funding. We really appreciate the support from Blood Cancer United. At Caribou, we've been developing off-the-shelf allogeneic CAR T cells for lymphoma and multiple myeloma. These are diseases where we know auto CAR T work, but the vast majority of patients actually can't access these innovative therapies due to either medical reasons, because they can't wait the weeks to months that it takes to apheresis and manufacture CAR T cells from their own T cells, or due to logistical, geographic, or financial reasons. It's a big struggle for patients to travel to the sites that are able to manage the logistical complexity of the apheresis and manufacturing of autologous CAR T cells. And this results in 75% of the large B-cell lymphoma patients and 90% of the multiple myeloma patients do not receive these potentially life-saving technologies. And so Caribou has been developing allogeneic off-the-shelf CAR T to try to solve some of these problems. And as you mentioned, I've been in this CAR T development space for a long time, and I've seen this challenge play out upon approval of these transformational CAR T cell products. So I'm really proud to be at Caribou, where we're developing the next generation of CAR T cells that can solve some of these problems. Caribou has sophisticated ways to genetically engineer healthy donor T cells and order so that they be ready for when the patient needs them and where they need them. And they can receive lymphodepletion immediately upon being identified as a patient eligible for CAR T cells because the cells are already manufactured and in the freezer. We have next generation CRISPR-Cas9 technology that we call chRDNA technology, and this allows us to do complicated multiplex editing while maintaining genomic integrity of these cells. And with this chRDNA technology, we're able to armor the CAR T cells for improved functional persistence. Since allogeneic products are made from healthy donor T cells that are foreign to the patient, they must be enhanced with technology that either makes them more potent while they're there or makes them stick around longer. And we do this by making cells that are either resistant to T cell exhaustion or by cloaking them from the patient's immune system so they can't be cleared as quickly and will stick around and kill more tumor cells. Our lead program, Vispacel, is a CD19 targeted allogeneic CAR T cell product, and we've just released data at the end of last year that shows in phase 1 that we can produce outcomes on par with autologous CAR T cells in second-line large B-cell lymphoma. This is really exciting because a lot of people didn't think that an off-the-shelf CAR T could be as good as something made from the patient's own cells. And because it's off the shelf and we can manufacture it in advance, this means we can get CAR T cells to more patients due to the speed of delivery and also the ability to get this type of product out into sophisticated community centers and not just have CAR T cells be a niche therapy that can be given at academic centers. And so we make Vispacel from young, healthy donor T cells. We modify them to insert the chimeric antigen receptor that can identify the tumor cell. We also knock out a PD-1 gene that then allows the cells to be more potent and not exhaust as they start killing tumor. We've treated 84 patients with Vispacel and shown overall response rates that are above 80% and complete remission rates above 60%. And excitingly, we've seen more than half of our patients still in remission when we go out 2 and 3 years after a single infusion of donor T cells. And as I mentioned before, these data really show that allogeneic CAR T cells can lead to outcomes similar to auto CAR T. So we're working with the FDA to finalize a registrational strategy in large B-cell lymphoma patients. Recently, we also disclosed data from our other program, which is not as far along in the clinic, but is in the middle of phase 1. This is CB11. This is a BCMA-targeted allogeneic CAR T cell for multiple myeloma. And as I mentioned, 90% of myeloma patients cannot get the approved auto CAR Ts that are available. And so this could really change the lives of a lot of myeloma patients. And so we genetically engineer the same healthy donor T cells with a BCMA-targeted CAR. And in this product, we actually cloak the cells from the patient's immune system by partially removing HLA proteins that the patient's body would recognize as foreign. And then we insert a fusion protein that further suppresses the patient's immune system from clearing the CB11 cells. And this allows the cells to actually persist for more than a month. We've recently shown that in 12 patients at our recommended dose for expansion that we have response rates over 90% and over 3/4 of patients are in deep complete remission. And even with sensitive tests to look something called minimal residual disease, over 90% of those patients reached a low detectable disease or MRD negativity. So this is really very encouraging in heavily pretreated myeloma patients, and we intend to enroll more patients at this dose level to confirm these outcomes and really understand what our steps forward would be with this product as well for patients with myeloma.
Thank you, Tina. That is very encouraging. And I share your aspiration of how do we make these products accessible to all patients? I think sometimes people don't realize that 80% of cancer patients are treated outside of the academic centers. And so how do we make sure all patients have access? So I'm really excited about what you've shared with us thus far. Tell us a little bit more about the current trials that are now enrolling and what patients are being treated and how our audience can learn more about sort of these future trials.
Absolutely. So right now with Vispacel, as I mentioned, our phase 1 study has finished enrollment. We're in the process of talking to the FDA about the final details around a registrational trial, a phase 3 trial in second-line large B-cell lymphoma patients. And this trial will be both at academic centers and also getting out into these sophisticated community centers. Vispacel has a safety profile very much like Breyanzi, which we know we can give as an outpatient. And so this really lends itself well to these sophisticated hospitals that want to give CAR T cells, but don't have all the logistical, set up to do both apheresis. And it really allows patients to get immediate treatment. So we're excited right now. We're focusing on getting those sites engaged and trained up to do CAR T cells. And many of them have already started doing treatments like bispecifics, which will make these sites very suited to know how to, use CAR T cells and treat or manage the toxicities that can come along with CAR T cells. And so for this program, we aren't enrolling patients right now, but we hope later this year to be doing so. And that trial will be listed on clinicaltrials.gov and the Caribou website. With CB11, we are continuing our phase 1 study at the recommended dose for expansion in patients that are triple refractory. So they need to have failed 3 lines of therapy. So very late line patients. And that will allow us to more fully understand the safety and efficacy of CB11 and understand then what the next steps might be to get that program into a registrational trial in the future to get approval and access to, again, another off-the-shelf allogeneic CAR T cell for patients. And similarly, you can look on the Caribou website. There's details about those trials and clinicaltrials.gov, of course.
Well, thank you so much. Kind of as we close out this discussion, which has just been terrific to learn more about this, maybe just highlight again, what kind of data, what will we be seeing in 26 or early 27, and what should we be looking forward to?
So this year, we will be continuing to follow all of our 84 patients that have been treated with Vispacel. I can assure you that we will be at multiple medical meetings this year and presenting these data in more detail and with more follow-up. This is the year where most of our patients will get out into that 1 year of follow-up with this drug, which is really exciting because with aggressive lymphoma, when you get out that far, there's a really high probability patients will stay in long-term remission, which is, of course, every oncologist and patient's goal in cancer care. So we're really excited that, the majority of our patients are still in remission. So we will be presenting those data later this year. Additionally, when we get higher numbers of patients treated with CB11 at the recommended dose for expansion, that also will give us a better estimate of efficacy and safety with that product as well. So you'll see data from that program over the year as well.
We will stay looking for that and excited to see this, how it plays out in particular. Hopefully patients will continue to have persistence and long remission times. And so we hope the best for them. Thank you again, Tina, for your comments. And I'd like to now ask that we bring back our other 2 speakers, Jan and Patri. And I'll turn it over to Blaine to have our Q&A.
Thanks a lot, Carol. Thanks, everyone. Welcome back, panelists. So thank you, audience, for watching. So we've just heard about a wide range of approaches that our biotech partners are developing for the next generation of therapies for blood cancer patients. So now looking at the Q&A panel, let me see what we have. So I think this question might relate to you, Jan. I know you didn't get a chance to sort of explain, your OncoMix platform, maybe possibly taking it beyond follicular lymphoma. The question is, anything that you're looking at for mantle cell lymphoma?
So what we think about is actually the similar setting as the watch and wait for follicular lymphoma. There are some patients also with mantle cell lymphoma that actually today are put in the watchful waiting category, where we think we could apply EU-2463 as well. So this is what we would like to start a study, an exploratory study to start with, of course. So that's one thing we would like to test. There are other indications as well, as with B cells, where patients also are put into in a watchful waiting situation where it could fit. But the next step, for me, it would be mantle cell lymphoma. I'm most interested in that one.
Wonderful. Thank you. I don't see any other open questions. We're almost out of time, but I mean, I'll open it up to any panelist. If you want to provide any closing comments or something, very briefly you didn't get to mention during the panel. Maybe Petri?
Thank you, Blaine. Really been a pleasure to participate in this discussion, and really want to thank once again Blood Cancer United and TAP to help Faron to develop BEX in MDS. It's been great in providing help in getting visibility in medical funding and industrial community for non-US company, and really to identify the KOLs and hospitals where to do our trials. And finally, also, it's been very important to have Blood Cancer United as a strategic investor at Faron. So really appreciate that, and it's been great to work together and really try to make everything to get BEX to patients as fast as possible.
Yeah, thanks. Tina, any closing comments? I know you just spoke, but
yeah,
anything
thank you.
Just
similarly, I think we're very thankful for Blood Cancer United. It takes it takes a village to develop these drugs, and we're really appreciative of your interest in Caribou and our next generation CAR T cell programs.
Great, thank you. Jan, I'll let you with any final words before we close out.
I think we have heard about three fantastic compounds that can change a lot for patients. I am getting very excited by the others speaks as well. Then, of course, I am so engaged in the oncomimics that this is what I want to work with. But I would echo what the others said, that the Blood Cancer United support it's not just funding, it's not just visibility, it's the knowledge that you carry with you and our regular meetings. I think this is a fantastic aspect. And all your contacts, this is extremely important for these smaller biotech companies. So thank you.
Yeah, thank you. I think we'll move to closing this out. So let's thank the panelists again for a great discussion and their very thoughtful answers. So as a reminder, this event's been recorded, so we'll be sharing it along with the panelists' contact information if anyone would like to follow up them specifically, and any additional questions that come to mind afterwards. So I again, thank everyone for watching today. I hope this event has really give you some food for thought about what we're doing here at Blood Cancer United, and some real optimism about some of these new therapies that are in development for blood cancer patients. So we at Blood Cancer United, remain committed to helping all patients, and at TAP will continue to broaden our reach with companies and patients in and outside of the US. So you can meet us in person. We'll be at all the major conferences, both at US and Europe, and we'll be back in January 27 with another exciting panel discussion. So again, thanks everyone, and we'll close out the meeting. Take care. Bye.
Thank you. Bye.
Thank you.